Hello, students. Welcome to the second session of Biotechnology and its applications. Let's look into the biotechnological applications in medicine. The recombinant DNA technological processes have made immense impact in the area of healthcare by enabling mass production of safe and more effective therapeutic drugs. Further, the recombinant therapeutics do not induce unwanted immunological responses, as is common in case of similar products isolated from non-human sources. At present, about 30 recombinant therapeutics have been approved for human use the world over. In India, 12 of these are presently being marketed. Let's look into the role of biotechnology in the treatment of diabetes. Management of adult onset diabetes is possible by taking insulin at regular time intervals. What would a diabetic patient do if enough human insulin was not available? If you discuss this, you would soon realize that one would have to isolate and use insulin from other animals. Would the insulin isolated from the other animals be just as effective as that secreted by the human body itself? And would it not elicit an immune response in the human body? Now imagine if bacterium were available that could make human insulin. Suddenly, the whole process becomes so simple. You can easily grow a large quantity of bacteria and make as much insulin as you need. Insulin used for diabetes was earlier extracted from pancreas of slaughtered cattle and pigs. Insulin from an animal source, though, caused some patients to develop allergy and other types of reactions to the foreign protein. Insulin consists of two short peptide chains, chain A and chain B, that are linked together by disulfide bridges. In mammals, including humans, insulin is synthesized as a prohormone which contains an extra stretch called the C peptide. The prohormone needs to be processed before it becomes a fully mature and functional hormone. The C peptide is not present in the mature insulin and is removed during maturation into insulin. The main challenge for production of insulin using recombinant DNA techniques was getting insulin assembled into a mature form. In 1983, Eli Lilly, an American company, prepared two DNA sequences corresponding to A and B chains of human insulin and introduced them in plasmids of E. coli to produce insulin chains. Chains A and B were produced separately, extracted, and combined by creating disulfide bonds to form human insulin. If a person is born with a hereditary disease, can a corrective therapy be taken for such a disease? Gene therapy is an attempt to do this. Gene therapy is a collection of methods that allows correction of a gene defect that has been diagnosed in a child or an embryo. Here, genes are inserted into a person's cells and tissues to treat a disease. Correction of a genetic defect involves delivery of a normal gene into the individual or embryo to take over the function of and compensate for the non-functional gene. The first clinical gene therapy was given in 1990 to a 4-year-old girl with adenosine deaminase deficiency. This enzyme is crucial for the immune system to function. The disorder is caused due to the deletion of the gene for adenosine deaminase. In some children, ADA deficiency can be cured by bone marrow transplantation. In others, it can be treated by enzyme replacement therapy, in which functional ADA is given to the patient by injection. But the problem with both of these approaches is that they are not completely curative. As a first step towards gene therapy, lymphocytes from the blood of the patient are grown in a culture outside the body. A functional ADA complementary DNA is then introduced into these lymphocytes, which are subsequently returned to the patient. However, as these cells are not immortal, the patient requires periodic infusion of such genetically engineered lymphocytes. However, if the gene isolate from marrow cells producing ADA is introduced into cells at early embryonic stage, it could be a permanent cure. Let's see what is molecular diagnosis. For effective treatment of a disease, early diagnosis and understanding its pathophysiology is very important. Using conventional methods of diagnosis, early detection is not possible. Recombinant DNA technology, polymerase chain reaction, and enzyme linked immunosorbent assay are some of the techniques that serve the purpose of early diagnosis. Presence of a pathogen is normally suspected only when the pathogen has produced a disease symptom. By this time, the concentration of pathogen is already very high in the body. However, very low concentration of a bacteria or virus at a time when the symptoms of the disease are not yet visible can be detected by amplification of their nucleic acid by PCR. Let's watch this video to understand more about PCR.
PCR has thousands of uses, like in diagnosing infections and infectious diseases. For example, it is used to diagnose infections by viruses. We take blood of a patient that has been infected by the virus, and then we amplify the DNA of the virus so that we are able to study which type of virus it is and its properties. Similarly, PCR is being used in hundreds of other fields like crime investigations, genetic research, molecular biology, etc. Now let's first talk about what are the things that we need to perform PCR, and then we will discuss how this process happens. The most commonly used equipment in PCR is the thermal cycler, also known as the PCR machine. Inside the PCR machine, we have these small tubes in which all the chemicals are inserted and the reaction takes place. Now let's look at the stuff that we put inside these tubes that makes this reaction possible. The key ingredients of a PCR reaction are Taq polymerase, primers, DNA templates, and the nucleotides. Taq polymerase is a type of DNA polymerase, and like DNA replication in any organism, PCR requires a DNA polymerase enzyme that makes new strands of DNA using existing strands as templates. The DNA polymerase typically used in PCR is called Taq polymerase after the heat-tolerant bacteria from which it was isolated, named Thermus aquaticus. This bacteria lives in hot springs, and its DNA polymerase is very heat stable, and it is most active around 70 degrees. This heat stability is ideal as high temperatures are needed for this reaction. The second important thing that we need to perform PCR are the primers. If you have watched the videos on DNA replication, you know that DNA polymerase needs primers to start the reaction, as the polymerase cannot initiate this reaction but can only propagate it. PCR primers are short sequences of nucleotides, usually around 20 nucleotides in length. Primers provide a starting point for DNA synthesis. Primers are also important as they help to select the exact portion of DNA that will be amplified. And we use 2 such primers in each PCR reaction, and they are designed so that they isolate the target region of the DNA to be copied. DNA template is the segment of original DNA which we want to amplify. And nucleotides, as we already know, are the basic building blocks used for DNA synthesis. Now we will discuss about the process of PCR. PCR involves 3 simple steps: denaturation, annealing, and extension. In the first step of polymerase chain reaction, we raise the temperature of the machine to 96 degrees Celsius. This is done to denature or separate the two strands of DNA. So, as a result of this step, we get two separate strands of DNA. The next step of PCR is known as annealing. Before getting into the details, do you know the meaning of the word annealing? Basically, this word was used in the metal industry, where they heat a certain metal to a higher temperature and then cool it. This process is called annealing, which is basically done to remove the internal defects from the metal. Same is the case here in PCR, where we first raise the temperature of the machine to 72 degrees for the step 1, that is denaturation, and then we cool the temperature to 55 degrees so that the primers in the PCR tube can bind to their target sequence on the single-stranded DNA. Now, let me explain the concept of annealing with a real-world example. Suppose a patient comes to you and you suspect an HIV infection. We take the blood sample of the patient and perform PCR on it. The blood of the patient contains the virus, and in the virus is its genetic material, the DNA. So our aim here is to confirm whether the patient has the infection or not. But the DNA of the virus in our sample is a very little quantity, so you can say that we need to amplify the DNA of the virus to detect it. So we perform the first step of PCR, that is denaturation, to separate the two strands of the DNA. This is the denatured DNA of the virus in our sample, and in green we have a specific sequence or gene that we have decided to amplify and detect later. Next comes annealing and the role of primers. We will use two primers which are basically used to mark the specific area of the DNA that we want to amplify. The primers have the sequence that matches the sequence which is present at the starting point of the target region of these two strands of DNA. The primers bind to the template DNA by complementary base pairing. In this way, we are able to select a particular region of the DNA that we want to amplify. So this was annealing. Next comes the third step, known as extension. For this, we also require free nucleotides in these test tubes, which will be basically used to make the new DNA. Again, we have a detailed video about the structure of nucleotides on our channel, and link is in the description. For extension, we increase the temperature again to 72 degrees so that the Taq polymerase extends the primers, synthesizing new strands of DNA. Here you can see the Taq polymerase adding new nucleotides to the short sequence of the primer to form new strands of DNA, which is complementary to the original strand. These ingredients are assembled in a tube along with cofactors needed by the enzyme and are put through repeated cycles of heating and cooling that allows new DNA to be synthesized. So what was the result of this single cycle of heating and cooling and then heating again? You can see that from one DNA fragment we got two new double-stranded DNAs. One is the original parent DNA and the other one is a new DNA formed by the Taq polymerase. These steps are repeated again, and this is what that makes the PCR a chain reaction. Basically, the products of the first reaction are used as the substrates for the next reaction. Now when we cool down the temperature to 55 degrees for annealing, we will get 4 single-stranded DNAs now. The Taq polymerase comes in for extension, and this time it leads to synthesis of 4 new single-stranded DNAs because the substrates for the enzyme are also double now. So from the second cycle of the PCR, we get 1, 2, 3, 4, 5, 6, 7 and 8 new strands of DNA. These steps are repeated again and again, and this is what that makes the PCR a chain reaction. To summarize, basically the products of the first reaction are used as the substrate for the next reaction, which means that if in the first reaction we got 2 DNA out of 1, in the second reaction we will get 4 DNAs out of 2. And in the third reaction we will get 16 out of 4, and similarly by a few hundred reactions we are able to get billions of copies of the same fragment of DNA. This is repeated 25 to 30 times in a typical PCR reaction, not 100 times, which takes 2 to 4 hours depending upon the length of the DNA region being copied. Due to the exponential nature of the chain reaction, we are able to produce billions of copies of DNA fragment from only a single copy of the fragment. After this, the temperature is again lowered to 15 degrees so that the products of the reaction can be stored. To check whether the PCR has generated the correct products, we use a technique known as agarose gel electrophoresis.
PCR is now routinely used to detect HIV in suspected AIDS patients. It is being used to detect mutations in genes in suspected cancer patients too. It is a powerful technique to identify many other genetic disorders. A single stranded DNA or RNA tagged with a radioactive molecule is allowed to hybridize to its complementary DNA in a clone of cells, followed by detection using autoradiography. The clone having the mutated gene will hence not appear on the photographic film because the probe will not have complementarity with the mutated gene. Enzyme linked immunosorbent assay or ELISA is based on the principle of antigen antibody interaction. Infection by pathogen can be detected by the presence of antigens like proteins, glycoproteins, etc., or by detecting the antibodies synthesized against the pathogen. In this session, you have learned about the role of biotechnology in the field of medicine. With this, we come to the end of today's session. Thank you. Have a pleasant day.